Did you know that Gregor Mendel cultivated and tested about 29,000 pea plants between 1856 and 1863? One such test was the crossing of pea plants with yellow and round seeds and those with green and wrinkled seeds. As you know, such a cross between plants that differ in two traits or characters is known as a dihybrid cross. This experiment, conducted by Mendel, resulted in a pea plant that produced yellow and round seeds in the F1 generation. Here, can you guess the dominant shape and color? Obviously, the round shape is dominant over the wrinkled shape and the yellow color dominant over green. Mendel noticed that these results were identical to the results he had obtained when he had carried out monohybrid crosses between plants with round and wrinkled seeds and those with yellow and green seeds. Now, in Mendel's dihybrid experiment, let's denote the dominant yellow seed color with the genotypic symbol capital Y and the recessive green seed color with small y. Likewise, let's denote the round seed as capital R and the wrinkled seed as small r. The genotype of the parents can therefore be denoted as capital R, capital Y, and small r, small y, small y. On crossing the plants, the gametes capital R, capital Y and small r, small y unite to produce yellow and round seeds with the genotype capital R, capital Y, small y in the F1 generation. Mendel further self-pollinated the plants obtained in the F1 generation to produce F2 generation plants. This time, he found that 3/4 of the seeds were yellow, while 1/4 was green. Similarly, he noticed that 3/4 of the seeds were round, while 1/4 was wrinkled. Therefore, the yellow and green color had segregated in the ratio 3:1, similar to the results obtained in a monohybrid cross. And the round and wrinkled shape had segregated in the same ratio 3:1. Mendel also found that the phenotypic ratio in the F2 hybrid was 9:3:3:1. That is, there were 9 round yellow seeds, 3 round green seeds, 3 wrinkled yellow seeds, and 1 wrinkled green seed. Mendel had got the same phenotypic ratio in several dihybrid experiments that he had conducted. This ratio 9:3:3:1 can be derived as a combination series of 3 yellow to 1 green with 3 round to 1 wrinkled. And further, this derivation can be written as shown. From such observations on dihybrid crosses, Mendel proposed a new set of generalizations which was later called the law of independent assortment. According to this law, when 2 pairs of traits or characters are united in a hybridization experiment, the segregation of one pair of characters is independent of the other pair of characters. We can use the Punnett square to easily understand the independent segregation of the 2 genes during meiosis and the production of pollen grains and eggs in the F1 generation. For example, in the dihybrid cross experiment conducted by Mendel, the genotype of the seed obtained in the F1 generation was capital R, small r, capital Y, small y. Now let's consider the segregation of one pair of genes, capital R and small r. Here, 50% of the gametes have the capital R gene, and the remaining 50% have the small r gene. Now, apart from each gamete having either capital R or small r, it must also have either capital Y or small y. Here, it is important to note that the segregation of 50% capital R and 50% small r is different from the segregation of 50% capital Y and 50% small y. Hence, 50% of the gametes bearing small r has capital Y, and the remaining 50% has small y. Similarly, 50% of the gametes bearing capital R has capital Y, and the remaining 50% has small y. Therefore, the genotypes of the gametes are of four types, namely capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. In other words, there are four types of eggs and four types of pollen. And as you can see, the frequency of these gametes is 25% or 1/4 of the total number of gametes produced. Now, the allelic composition of the zygotes of the F2 plants can be easily obtained using the Punnett square as shown. As you already saw, there are four different types of phenotypes formed, and the phenotypic ratio is 9:3:3:1. However, 9 different genotypes result from the F2 hybrid, and the genotypic ratio is 1:2:1:4:2:1:2:1. Thus, from the dihybrid cross experiment, Mendel found that the gene combination of the progeny differs from the parental gene combination.